Hi guys, welcome back to another episode of Canberra's Guide to Not Effing Up A-Level Biology. Today we're going to be doing the control of the heart rate. This is effectively an extension of the typical heart stuff you did in year one. It's going to have a bit more nerve impulsy stuff in it because it's a lot more complex and we actually go over the pacemaker cells that are involved in regulating heart rate. So this is it's a pretty fun one in my opinion. So let's get started. So the best thing to do is starting off by drawing a little heart, and you should know how bad I am at drawing hearts now. Well, I wasn't good in the first place, but anyway. So I'm gonna draw a very ugly heart here. I'm just gonna draw the 4 chambers. Obviously this is wrong because each chamber is enclosed when it's not supposed to, but just get the general gist of what's going on. Okay, so over here we have something called prochyne fibers, and prochyne fibers they're effectively like muscles you can think of. They effectively carry the electrical impulses throughout the heart, so you can effectively pump blood around the body. So prochyne fibers, and a bunch of these prochyne fibers make up something called the bundle of His. So I'm just going to draw a few more there, and we call that the bundle of His. The corner over here, so the furthermost part of the heart, we call that the apex. And as you can see over here, the prochyne fibers, they branch off along the walls of the the ventricular muscles over here and here. And that's effectively to cause proper contraction, ensuring that all of the ventricles contract properly and that you can have that pressure change within the heart. In another colour, I'm going to put over here the pacemaker cells, which effectively help regulate heart rate. And over here, we have something called the atrioventricular node. I'm going to call that AVN. And we're going to go over what the AVN does now really, because we're going to go over how a normal heart contraction happens. So the two things that we should start off by going over is the sympathetic and parasympathetic nervous systems. Pathetic. Parasympathetic. Sympathetic, you can think of your body almost being nice to you. It's effectively there to stop you from doing something dangerous. Your body is kind of looking out for you. And what it effectively does, like the biological version of what it does, is that it increases nerve impulses to effectors. So say you're in a really stressful situation and you're being killed. Your body's gonna, your sympathetic nervous system is going to increase impulses to your effectors, such as your adrenal glands, to increase the amount of adrenaline released in your body. So that gives you that fight or flight response, which increases your rate of survival. That's why I like to think of it as being called sympathetic, because your body's actually looking out for you. Whereas parasympathetic is essentially the opposite of it. So it reduces or inhibits nerve impulses, nerve impulses to effectors. So you say like you somehow manage to escape and you kind of need to calm down, there's not much of a risk anymore. Your parasympathetic nervous system will kick in and it's going to start inhibiting those nerve impulses to the effector. So like your adrenal gland, for example. One thing you need to know is that they almost work against each other. I like to think of it as like antagonistic muscles, because one does something, the other doesn't. So like if I'm gonna like move my muscles, I don't have muscles. But if I'm going to move my hand up or down, the muscles inside those hands, one of them is going to contract, the other is going to relax, and vice versa. So that's what we mean by antagonistic. And that's pretty self-explanatory if we look at the sympathetic and parasympathetic nervous systems over here. Alright, so let's go over the actual mechanism of a heart contracting. At GCSE level, we learned that pacemaker cells regulate heart rate, and yes they do, but there's a lot more to it at A level. So the first step that happens is the pacemaker cells. In A level, I shouldn't actually be using the word pacemaker cells, I should be using the word sinoatrial node. So I'm going to replace pacemaker with SAN. It's just another fancy word for pacemaker, so it's fine. So, first step is this SAN initiates a wave of electrical excitement I'm going to say electrical with the electric, like electron symbol. Electrical excitement along the atria. So over here you have your SAN, and it's going to spread a wave of electrical activity to the ventricles here. I should have used another colour, sorry about that. There we go, black. So let's say the black is the wave of electrical excitement. Now, electricity causes muscles to contract. We should have known that from the muscles topic. So all of that electrical activity, that's causing the ventricles, always get messed up, the atria to contract. So I'm going to write that down. Which causes both atria to contract, pumping blood into the ventricles. And the key words here, obviously SAN, initiates wave of electrical excitement, and that causes the atria to contract. Those are the main things you need to write down. So that's the first step. The second step is that the wave of electrical excitement cannot reach the ventricles due to the atrioventricular septum. This is where it gets a bit weird, but the atrioventricular septum is basically some non-conductive tissue, and it's found over here. And it essentially stops that wave of electrical excitement from passing through it. The main reason is to essentially help the ventricles fill up with blood. If they all started contracting at the same time, it wouldn't be a very efficient system of pumping blood around the body. So effectively what happens is that because you're stopping that wave of electrical activity going down, you're giving the ventricles a chance to fill up with blood. Instead, what happens is that that wave of electrical activity reaches the AVN, which we talked about. We haven't really talked about, we're going to talk about now. So, I think with here, it's good to actually kind of describe the atrioventricular septum. So, non-conductive tissue that prevents wave of electrical excitement reaching the ventricles. Rather, it reaches the AVN, or the atrioventricular node. Now, the AVN is kind of the secondary pacemaker, the way I like to think of it. So the first assay, so the sinoatrial node, it's done its job, it's passed on that wave of electrical activity, and that's when, after the AVN does the second part of heart contraction, which is to contract the ventricles. So what we're going to say next is that after a short delay, the AVN, it basically conveys that signal. So that after a short delay, the AVN initiates a wave of electrical activity or excitement, same thing, to the Purkinje fibers, which make up the bundle of His. There we go. Alright, so we have this electrical activity flowing down the Purkinje fibers. The next thing it does is that as it's being signalled along those Purkinje fibers, it will reach the apex of the heart, or the corner of the heart. So I think just to save some time, because you don't want to write wave of excitement over and over again, it's going to take a lot of time, you can say which makes up the bundle of His, whereby this activity reaches the apex of the heart. The next thing we write is after it reaches the apex of the heart, the wave of electrical activity reaches the ventricular muscles via the Purkinje fibers. So you can see that it's very repetitive. You don't have to write it over and over again. But essentially, this is what will get you the marks. You're just really solidifying what you're trying to say. So the wave of electrical activity reaches the ventricular muscles via Purkinje fibers. This causes the ventricular muscles to contract, which pumps blood out of the heart. Alright, so those are the key points. Let's do a bit of highlighting so we know what we're supposed to write in the exam. So let's start from the beginning. The sinoatrial node initiates a wave of electrical activity along the atria, which causes both the atria to contract, pumping blood into the ventricles. Alright. The wave of electrical excitement cannot reach the ventricles due to the atrioventricular septum, non-conductive piece of tissue that prevents the wave of excitement from reaching the ventricles. Rather, it reaches the atrioventricular node. After a short delay, the atrioventricular node initiates a wave of electrical activity to the Purkinje fibers, which makes up the bundle of His, whereby this activity reaches the apex of the heart. The wave of electrical activity, well, I'm writing slowly down. The wave of electrical activity reaches the ventricular tissues, or ventricular muscles via Purkinje fibers. This causes the ventricular walls to contract, which causes blood to leave the heart, or being pumped out of the heart. So that's essentially how electrical activity works in the heart. The last thing that you need to know is how we actually control heart rate, how the body kind of manipulates the system in order to ensure that we have enough oxygen flowing through the body. So for example, like if you're doing some exercise or something, you'll realize that your heart is beating a lot faster. And there is a mechanism involved to it, which we're going to look at now. So what I'm going to do is, I'm going to just draw a brain. I'm going to rub this out because I think we've kind of got the general gist of what's going on. There we go. So, draw the brain in red. I know, ugly brain. And then we're going to draw a part of the brain. And this is called the medulla oblongata. It's probably in the wrong position, but you don't need to know about that. So, medulla oblongata. And the medulla oblongata is responsible for controlling the heart rate of your body. And the mechanism we're going to we're going to go over, it's going to start like this. So let's say you go exercising and you start encountering an oxygen dip. So let me think. So yeah, let's go along with the scenario. So you have an oxygen depth, so carbon dioxide levels in your blood increases. So CO2 levels in blood increases. Now from year one, you should know that carbon dioxide in the blood can dissolve, and that can form carbonic acid. So CO2 dissolves in blood to form carbonic acid, which lowers the pH. Now, by lowering the pH, the body will detect that, and as you know, with homeostasis, it's the establishment of conditions that are balanced, that allow the body to do its metabolic processes in an appropriate way. So we have some things that will detect that change in pH, and those are called carotid arteries. So I'm going to draw an artery there. So carotid artery, and they contain chemoreceptors that will detect that change in pH. So carbon dioxide dissolves in the blood to form carbonic acid, which lowers the pH. So the next point we're going to write is that this change in pH is detected by chemoreceptors in the carotid arteries. What this does is that by detecting that change, those chemoreceptors start sending more frequent impulses to the medulla oblongata, and by sending those more frequent impulses, the medulla oblongata will start sending impulses to the sinoatrial node, again at a higher frequency, to ensure that the heart beats a lot faster. Now, if I'm going to be entirely accurate, it's not the medulla oblongata itself. There's a center in the medulla oblongata. Medulla oblongata, I'm going to draw that as a dot. So I'm going to say center over here, because that's technically what we should be referring to when we're dealing with this mechanism. So this change in pH is detected by chemoreceptors in the carotid arteries. This causes chemoreceptors to send an increased frequency of impulses to the center in the medulla oblongata, responsible for controlling heart rate. After that, the center, so I'm going to say this center, now sends an increased frequency of electrical impulses to the sinoatrial node, which we know is the pacemaker cells, which causes the heart to beat faster. So your increased heart rate, and as heart rate increases, more CO2 removed from blood. And that is an example of negative feedback. So we saw this in the case of blood CO2 levels increasing. In the case that it decreases, basically the opposite would happen, and your parasympathetic nervous system will start kicking in, Just to make sure, this is part of the sympathetic nervous system, because what we're doing is we're stimulating effectors and increasing the frequency at which we're stimulating them. So let's go over this one more time. As you know, I like to highlight the things that will get the marks. So firstly, CO2 levels in the blood increases. CO2 dissolves in blood to form carbonic acid, which lowers the pH. This change in pH is detected by chemoreceptors in the carotid arteries. This causes chemoreceptors to send. My handwriting is really bad. This causes the chemoreceptors to send an increased frequency of impulses to the center in the medulla oblongata responsible for controlling heart rate. This causes this center now sends an increased frequency of electrical impulses to the sinoatrial node, which causes the heart to beat faster. As heart rate increases, more CO2 removed from the blood, and this is an example of negative feedback. Alright, so that's all of control of heart rate done. I think the next video we're going to do is... What am I going to do? I think it's going to be muscles again. But nevertheless, I hope that was very helpful. I'll see you in the next video.